Okay, let's continue on chapter 14, diversity and macroevolution. And I hope we'll be able to finish this chapter with this video lecture. So this should be the last video lecture of the semester. Okay, we looked at tectonic plates, continental drift. As I said, sometimes in science, you got to have an open mind. You know, when this guy first, Alfred Wegener, suggested this, people thought he was crazy, but he was right. Continents move over time. So it has a tremendous impact on the evolution of life's diversity, reshaping the physical features of the planet and altering environments in which organisms live. Antarctica used to be a tropical environment. They were like fern fossils down there, but now it's a frozen wasteland. Okay. 250 million years ago, plate movements formed a supercontinent Pangaea, reducing the total amount of shoreline, deepening ocean basins, lowering sea levels. So here, Pangaea has formed a supercontinent, and this is where these continents moved apart. It splits into Laurasia and Gondwanaland. Only had these, and then India collides with Eurasia, producing the Himalayan mountains. Okay. And the sliding of these plates against each other causes earthquakes. Okay, second dramatic chapter, history of continent drift began in the mid-Mesozoic area. Pangaea started to break up, causing geographic isolation, colossal proportions. And as they drifted apart, each continent became a separate evolutionary arena as climates changed and its organisms diverse. And that's actually during the dinosaur time of the dinosaurs, the Mesozoic. So this shows patterns of biogeography, study of the past and present distribution of organisms. Continental drift separated Australia from other land masses, and they protected marsupials. Marsupials are pouched mammals, and they're really found nowhere else in the world, except before this checkpoint. If marsupials originated in Asia and reached Australia versus by South America, where else should paleontologists find fossil marsupials? Well, in South America. That's where marsupials started. They went to Australia from South America, and South America at one time was isolated too, an island continent. But then it got connected to North America, and so-called eutherian mammals, placental mammals, came down across Central America and they outcompeted marsupials. They wiped out marsupials. And this is why our marsupials still exist in Australia, because it was isolated, because it's an island continent. One exception to that wipeout of marsupials when eutherian mammals went down from North America, opossums, they were really, really successful, and they went in the opposite direction, and they populated North America from South America. And they're the only marsupial we have up here. So there's some Australian marsupials. Okay, I mentioned before 5 mass extinctions over the past 540 million years. In each of these events, 50% of more Earth species died out. So it's the Permian, 96% of marine species, Cretaceous extinction, about 66 million years ago, that caused the extinction of that helped the extinction of dinosaurs, except for birds, remember. Birds are pretty close to living dinosaurs. Extinctions seem to provide surviving organisms with new environmental opportunities. For example, so prior to the Cretaceous extinction, our ancestors were these tiny little like rodent-like things hiding at nighttime from these big, terrible lizards that are walking all over the place, the dinosaurs. And you know, for example, the plains were filled in North America with herbivorous dinosaurs that would eat grass. Okay, dinosaurs get wiped out. We have open environments, resources that aren't being used. So mammals moved in to where those dinosaurs were in these ecological niches and took over. So then we have bison, American bison, huge herds of those, and antelopes, mammals now going over and eating grass in the Midwest, replacing the dinosaurs that used to do the same thing. So that was called an adaptive radiation. So that provided opportunities for for new organisms, the mammals, to come in and take the place of dinosaurs. So there's reptilian ancestor, ancestral mammal, and this is diversification of mammals. There's three types of mammals: monotremes, which are really primitive mammals that are actually living fossils between reptiles and mammals. They're egg-laying mammals. Then there's the marsupials, pouched mammals, and then there's the majority of mammals are called eutherian mammals. They're placental mammals. After the extinction of the dinosaurs. Okay, so scientists are increasingly able to explain the basic biological mechanisms of macroevolutionary changes seen in the fossil record. Scientists have tell how slight changes in the flow of genetic information can become magnified into major structural differences. And as I said, we all have these basic genes that tell us where to put arms and legs and heads. And so the basics, those are all the same for all these organisms. But during embryonic development, other genes start modifying those things into very, very diverse ways. Okay, so evolutionary transformations can result from genes that alter the rate, timing, and spatial development changes in an organism's form as it develops from a zygote into an adult during embryonic development. These other genes change things. And even though we have all the basic genes are all the same between all the mammals, actually, and birds and reptiles as well, these genes change and make very different looking organisms. Okay, comparison of human and chimpanzee skull development. Here's the thing. This is called pedomorphosis, P-A-E-D-O-M-O-R-P-H-O-S-I-S. It's in the book. The retaining of juvenile characteristics as an adult. And it's thought that human beings actually underwent pedomorphosis from our ape-like ancestors. If you look at chimpanzee fetus and look at the skull, and then you look at chimpanzee adult skull, it looks really different. The brain case is smaller, it's a huge jaw and maxilla bone. Look at a human fetus and a human adult. They're not that different. So we seem to be retaining juvenile features in our skull. And this is a pedomorphic feature. We're pedomorphic apes. This is true for some salamanders too. Salamanders, when they're developing, they have gills. Same thing with frogs, have gills, and tadpoles. But eventually these gills are resorbed and they grow lungs, although some salamanders are actually lungless, and then they can go out on land. Some salamanders retain the gills and never develop lungs, and they stay in water all the time. And this is a pedomorphic feature. The gills are embryonic or adolescent thing, and they're retained as an adult. Okay, so of course a giraffe evolution increased growth rates during development produced extra long inch vertebrae. We have 7 vertebrae in our neck. How many vertebrae do giraffes have? Well, 7. All vertebrates, how mammals have 7 vertebrae in their neck. They're just a lot longer, all right? And this is interactions within biological systems. Feather-flighted birds are perfect marriage of structure and function. How did its intricate structure evolve? Thousands of fossils of feathered dinosaurs have been found, but those feathers weren't used for flight. They were thought, and I think I mentioned this before, to be thermal insulation first, and then they became adapted for flight. So there is Archaeopteryx, extinct bird, but it's got teeth like a reptile, claws and forms like reptiles, long tail of vertebrae like a reptile, but it's got feathers. It's a feathered dinosaur. So the first utility feathers may have been used for insulation. Once flight became an advantage, natural selection would have gradually modified feathers and wings to fit their additional function. So structures as feathers that evolve in one context, but become co-opted for another function are called exaptations. So thin membrane of skin stretched between long fingers of bones for bats is an adaptation for flight. Which major theme is this? Structured function. That and that could be also metamorphosis. As embryos, we have webbed fingers. So all the bats had to do to make wings is to maintain that webbing between the fingers. And then they made a wing out of it. Okay, so most complex structures evolve in small text. Simpler versions that have the same basic function, process of refinement, rather than a sudden appearance of complexity. So complex eyes can be traced from a simple ancestral patch of photoreceptor cells through a series of incremental modifications that benefit their owners at each stage. A lot of people say, what's good as half an eye? What's better than no eye? So real simple eyes are just light detectors, right? And then they became more sophisticated in trying to see images. Why does the concept of exaptation does not imply that a structure evolves in anticipation? Because it had one role, it started off with one role, and it wasn't anticipating flight, but it became adapted for flight afterwards. So the eye, patch of pigmented cells, it's just a light detector in like a lipid. Then we have an eye cup in the abalone, fluid-filled cavity in the nautilus, then a lens in a marine snail, and then complex cameral lens type eye, just like ours, in a squid. And with eyes, I want to also talk about something called design. And this is why the human eye is a really good example of how that intelligent design, which is a creationist idea, that we're designed really, really well, and that proves that there must have been some creator or something. No, human eyes are designed backwards. Here's the thing. In the ancestor of squid, these pigmented cells were pointed toward the light. And so squid have eyes that have the retina where the photoreceptors are pointing toward the light, and all the wiring is behind that. But in our ancestor that had the simplest form of eye, those cells were put backwards. They were facing away from the light. And then they started making more complex eyes in other ancestors. But the genes that laid down those photoreceptors are set in stone. So now our eyes are designed backwards. Our photoreceptors point away from the light. And so that's why we have a blind spot in our retina, because the ganglion cells whose axons make up the optic nerve, they have to collect and form the optic nerve and go through the retina into our brain. We can't put photoreceptors where the optic nerve is forming. So that's our blind spot. So we have a flaw in our retina because it's backwards. Does that mean our eyes don't really work well? No. Poor design doesn't mean things don't work really well. Our eyes work fantastically. They just have this little flaw because they're set up backwards. Why don't we have a little black space in our field of view? Because your brain fills that over. All right. It sees, I'm looking at this white wall. The brain sees white on one side of the blind spot. It sees white on the other side of the blind spot. So it just smooths it over and makes it all white. So this is design. Now so this is an intelligent design. What kind of an engineer would make a light detector with photoreceptors pointing in the wrong direction? So, and the only reason that they are like this because they evolved that way. And they work really well. You just have to, patch over that little flaw in the retina because it's backwards. Okay. Taxonomy, remember the naming and classification of species. Systematics include taxonomy, focus on classifying organisms, determining their evolutionary relations. We've talked about this before. So we make phylogenetic trees to depict hypotheses about the evolutionary history of species and reflect the classification of groups nested within more inclusive groups. So here's a family tree of carnivores. Order Carnivora, family Felidae, panthers, mustelid, otters and skunks, Canidae, dogs, coyotes, wolves. And so I'm saying phylogeny and practical applications. So constructing the phylogeny of maize, researchers have identified 2 species of wild grass that may be maize as old as living relatives. And they may have alleles that offer disease resistance, could be transferred into cultivated maize. And also, I think the ancient ancestor of maize is a perennial, as it comes up every year. Corn is an annual. It only comes up 1 year and then dies. So if you could transfer those genes, you could have corn that's a perennial, and the farmers don't have to go in there and cultivate their fields every year. It'll just come up all by itself every year. So homologous structures of different species. Remember, they're from common ancestors. May vary in form and function, but it's fundamental similarities because they evolved from the same structure of common ancestor. So homologous structures are one of the best sources for information for phylogenetic relationship. However, convergent evolution involves superficially similar structures from different evolutionary branches that result from natural selection shaping analogous adaptations. Similarity to the convergence is called analogy, not homology. Let me give you a good example. I think I gave this example before about sharks, which are fish, obviously, ichthyosaurs, which were actually marine dinosaurs, and I say dolphins. They all look superficially similar. They're all really streamlined. And why? It's because they are really fast swimmers in a thick medium, water. So they have to be streamlined to go fast. So they look, actually, if you look at them, they all look pretty similar, but they're all really distantly related to each other. And so this is analogy. This is not homology. The fact that, remember, we looked at forearms, we looked at legs and wings and arms in all the different types of mammals, and they all have the same bones. They're just arranged in different ways. Now that's homology from a common ancestor. Okay, comparing embryonic development, 2 species can reveal homology. So we've looked at that before, how all the embryos of mammals and birds look almost alike when they're really young. So if homology reflects common ancestry, then comparing the DNA sequences of organisms get to the heart of the evolutionary relationships. The more recently 2 species have branched from a common ancestor, the more similar their DNA sequences should be. We talk about this in the lab. So some fossils are preserved in such a way that DNA fragments can be extracted for comparison with living organisms. And there's art of construction of Neanderthal man. Okay, we're gonna skip by cladistics. I'm not getting into cladistics. We're not gonna do that. However, this is an interesting arrangement of reptiles and dinosaurs. So here are the crocodile, dinosaurs, crocodiles, lizards, and snakes. They broke off a long time ago. They're really distantly related. Crocodiles are really closely related to dinosaurs also. They're called archosaurs. So a common ancestor of crocodiles, dinosaurs, and birds. And then we got the two types of dinosaurs, which you don't have to know the different type of dinosaurs. These kind of dinosaurs and these kind of dinosaurs. And look how closely related they are to birds. Like I said, birds are pretty much living dinosaurs. Okay. Five leg trees are hypotheses about evolutionary history. Linnaeus divided all known forms of life between the plant and animal kingdoms. They just had plants and animals. That's it. And they actually, well, for one thing, they didn't know about bacteria. Then in the mid-1900s, a two-kingdom system replaced a five-kingdom system that placed all prokaryotes into one kingdom, used to be called Monera, and divided the eukaryotes among four other kingdoms. Then we went on to molecular studies, and ballistics led to the development of a three-domain system. Two domains of prokaryotes, bacteria and archaea, and one domain of eukaryotes, eukarya. And the domain eukarya is currently divided into kingdoms, but the exact number of kingdoms is still under debate because the kingdom Protista is like a real hodgepodge of all kinds of organisms. The only thing they really have in common is they're all unicellular eukaryotes, but actually not all of them are. There are actually some multicellular. So that's a real hodgepodge, and they may break that down into actually individual kingdoms later on. Okay, so and here, so here's the three-domain classification system. And here we show something that's really odd that was discovered by the analysis of DNA. This was not something that was obvious from just looking at the structure and function of these organisms. Because look at the evolutionary relationship. The earliest organisms broke up into a domain bacteria and this domain archaea and domain eukarya. What this thing is saying is we are more closely related to the archaea than the archaea are to domain bacteria, despite the fact that archaea and bacteria are both prokaryotic organisms. But so that's something that was new and was not anticipated when they finally looked at DNA. Our DNA and archaea DNA are more closely related than the archaea DNA to bacterial. Something that wasn't anticipated. Okay, so plants, bacterial, organic, from inorganic nutrients. Why is it bringing up photosynthesis here? I don't know. Pathways that transform energy and matter. So the Anthropocene, time of environmental change on an epic scale, we're affecting habitats, we're changing the face of the earth, and causing climate change, and causing the sixth mass extinction on Earth. Some organisms become resistant to toxic pollutants. Crested animals in Puerto Rico, sticky scales, adaptation to slippery surfaces. Urban blackbirds sing at higher pitch, adaptation that enables them to be heard over traffic noise. So they are adapting to the humanly altered environment. So urban adapted animals, they're the ones who are going to be the most successful organisms. And a lot of animals have to become adapted to human environments. A good example is what's the woodpecker I'm trying to think of? Oh, no, I can't think of it. It's a big, hang on a second. Pileated woodpecker, that's what I'm trying to get at. Are we still, okay, yeah. these pileated woodpeckers, these big woodpeckers with a big red crest, you used to only see them deep in the forest. You had to get really deep with. Now you're starting to see them in backyards. I just saw one riding to the HCC in my neighborhood. I saw a pileated woodpecker right in the neighborhood. So they are adapting to people environments. So they're probably going to make it. Okay, now the last thing I want to talk about is its latest transitional form, fossil found called tiktaalik. It's really exciting. So let me stop screen sharing this and screen share from Google about tiktaalik. Let me stop this show here. In show, okay. Okay, tiktaalik. It's actually an Inuit word because, well, here's the thing. The idea was that amphibians evolved from lobe-finned fishes. These lobe-finned fishes, they actually were thought to be completely extinct until some trawler in the 1930s pulled one up off the coast of Madagascar. They were around about 380 million years ago. And before that, they had lobe-finned fishes. And then around 360 million years ago, we had amphibians. And they looked like similar, but they still had very different structures, right? And so the idea was that amphibians must have evolved from these lobe-finned fishes, right? But, and that transitional organism must have lived about 375 million years ago. Between the 380 or 390 million years ago with lobe-finned fishes and the 360 million years ago with the first amphibians. So we're going to look at rocks that are around like 375 million years in between those. So they scoured the Earth trying to find exposed rock of that age. And they found it on Baffin Island, way up in the northern, in the Canadian Arctic. They found exposed rock of that. So they started looking. And lo and behold, so this is a prediction. We should find an organism that has fish-like features and amphibian features that lived about 375 million years ago. And there it is, right there, tiktaalik. It's called a fishapod, 375 million year old fossil fish. Its discovery shed light on a pivotal period of history of life on Earth when the first fish ventured out onto land. It's a cross between a primitive fish and four-legged animals. So it is the ancestor of the tetrapods, of all the tetrapods. Tetrapods are land-dwelling animals with four limbs, which includes us, and birds, because they have four limbs, just two of the limbs happen to be wings. And all of the mammals, all of the amphibians, all of the reptiles, mammals, birds, they're all tetrapods. And this is the great-great-great-great-grandfather of all tetrapods, the first four-legged animal to walk on land. And the incentive was, there was a bunch of stuff on land. Now, before the tetrapods walked on land, there were lots of insects on land, lots of plants, fungi and insects. No big thing was eating these insects. And so it was adaptive radiation. It was a chance of, to eat something that nobody else can eat. If I can get out of the water and walk out of the swamp and walk on land, I can eat these things. So tiktaalik lived about 12 million years before the first tetrapods. So the existence of tetrapod features in a fish like tiktaalik is significant because it marks the earliest appearance of these novel features in the fossil record. And what are we talking about? Well, one thing we're talking about is ankles. and wrists, ankles. Fish don't have ankles. This thing had primitive now, and the oldest amphibians had regular ankles. This thing had a real primitive ankle. Well, let's look. See Tiktaalik's mix of fish and amphibian features. Okay, so a traditional organism has combinations of features of the two organisms together. So Tiktaalik shares anatomical features with both primitive fish and the first tetrapods. It has features we really associate with fish: fins, scales, gills. But it also has a number of key features that differentiate it from its fishy ancestors, and that is head and body are flat with eyes on the top of its skull, like a crocodile. Its shoulders are not connected to its skull, giving it a functional neck, which fish don't have. And it has ribs, like some of the earliest tetrapods. And like I said, it doesn't mention, but primitive ankles. So it could actually, and actually, the only ankles it had were in the first two limbs. The back limbs were still fins. So it probably had to drag those, so it could walk with the two front legs and at least get onto land and start eating this stuff that no other animal was eating. Well, there's other insects that eat other insects, but no big animal was eating the insects. So could Tiktaalik walk on land? It's very unlikely that it walked entirely on land. Structure of its front fin and its shoulder, we know that it was capable of swimming and propping itself up. And probably so lived most of life in shallow water close to shore, using its strong front legs to push along the bottom and stabilize the moving water. But so it couldn't walk like an amphibian, but it could come close to shore and get onto shore in its really shallow water, and then but it could survive for strategy, convenient way to escape the huge carnivorous fish that live in the deeper waters, and it helped pursue some of the smaller fish and the bug-like critters along the dense foliage along the banks. So it was a really important fossil find of a truly transitional organism. And this page here mentions, and you can go on to the web and just type in Tiktaalik and pull this up, mentions how they first predicted that they should find it. How they had to find, they knew it was 375 million years ago, find rock and just go out and find it. And they did. It was remarkable. Okay. So that's it, folks. So this is 106 diversity 2, and so we're finished with chapter 14, and the material for the final exam. So I'll stop recording and see you later.